Antiviral properties of sunlight, Part 3, Ferroptosis inhibitors. This YouTube continues a discussion about the roles that solar radiation play in the production of two key immune hormones in the human body, calcitriol, 125 vitamin D3, and melatonin. We will connect this discussion to YouTube's on the MedCram channel, a case study, sunlight for hospitalized COVID patients, and a newly posted YouTube, new hospital prioritizes sunlight and fresh air. Prioritizing sunlight and fresh air once again, the new Footscray Hospital in Melbourne, Australia. Parts 1 and 2 of this discussion can be accessed from these URLs. The science of sunlight, why the new Footscray Hospital is built differently. Next Wednesday, that dark place, the old Footscray Hospital, will be replaced by the new Footscray Hospital, a $1.5 billion health service on the corner of Ballarat and Geelong roads that harnesses natural light to help patients heal faster. The design is backed by extensive research showing natural light helps patients establish a healthy circadian rhythm, leading to better sleep and a faster recovery time. Research has found that patients with beds near a window spend less time in hospital than those near a door, and those with access to high levels of natural light experience less distress. Light has also been found to prevent viral and staph infections and decrease a patient's heart and pulse rates. Besides improving the circadian rhythm, what other ways can sunlight improve the physiology of hospitalized COVID patients? In this YouTube, we will concentrate on a discussion of sun-induced ferroptosis inhibitors. These include melatonin, which is synthesized inside mitochondria in response to near-infrared wavelengths of sunlight, and two very powerful immune hormones, calcifediol, 25 vitamin D3, and calcitriol, 125 vitamin D3. They are downstream metabolites of vitamin D3, which is synthesized in human skin in response to ultraviolet light. On the right are several information vectors describing briefly how these immune hormones, melatonin, calcifediol 25 vitamin D, and calcitriol 125 vitamin D, can upregulate the master anti-inflammatory antioxidant transcription factor, NRF2, NRF2. NRF2 in turn upregulates a key antioxidant enzyme, GPX4, glutathione peroxidase 4. It is through the action of glutathione peroxidase 4, by utilizing glutathione, this enzyme is able to reduce reactive oxygen species as well as peroxidated lipids, which are upstream drivers of the programmed cell death known as ferroptosis. And ferroptosis is a key pathophysiological mechanism that is in many hospitalized COVID patients, as well as patients who have exited the hospital and have IAC conditions, infection-associated chronic conditions. In previous YouTubes, we have shown this figure, cumulative publications on ferroptosis. Since the COVID pandemic, approximately 23,000 papers have been published. Prior to the COVID pandemic, only 2,000 papers had been published on ferroptosis. This rapid surge in the study of ferroptosis is driven by the recent understanding that ferroptosis is a key driver of many progressive and chronic conditions. If you go to PubMed and search for melatonin and ferroptosis, you'll find 132 results. Some key references to draw from. Therapeutic effects of melatonin in the regulation of ferroptosis, a review of current evidence, published in 2024. Melatonin, a ferroptosis inhibitor with potential therapeutic efficacy for the post-COVID-19 trajectory, in other words, long COVID. The post-COVID-19 trajectory of accelerated brain aging and neurodegeneration. Role of ferroptosis in the progression of COVID-19 and the development of long COVID, 2025. We will concentrate on this paper, critical role of ferroptosis in viral infection and host responses. This figure has a number of major attributes. In contrast to other physiological diagrams, which often employ curvilinear lines to connect enzymes and transcription factors, metabolic and epigenetic events, this paper uses more of a block diagram, which is familiar to many technical professionals, such as computer scientists and engineers. It illustrates a flow diagram looking at metabolic and signaling pathways which are involved in the production of the cell behavior known as ferroptosis, a type of programmed cell death which is triggered by a dysregulation of iron. However, in this diagram, the elements are working in parallel to iron dysregulation, and they involve aberrations in the synthesis of glutathione. GSH is the abbreviation for that. We will take a look at this diagram in the context of it being a metabolic map of viral ferroptosis. I'm adding some additional agents to connect this diagram with other papers. GPX4 acts on reactive oxygen species as well as phospholipids that have been peroxidated. These are known as phospholipid hydroperoxides, and that's abbreviated PLOOH. In other papers, these are referred to as lipid hydroperoxides, and those are abbreviated as LOOH. Reactive oxygen species, that abbreviation is ROS, ROS. In discussing this metabolic map of ferroptosis, it's important to put ROS and peroxidated lipids downstream of GPX4, because GPX4, by utilizing glutathione, is able to chemically reduce reactive oxygen species into neutral molecules. The same is true for peroxidated lipids. GPX4 chemically reduces these peroxidated lipids, which are upstream drivers of ferroptosis. So it is possible to conceptualize this map as a folder of information on viral ferroptosis. Note that the authors have delineated a set of viral actions using a dotted line. These are on the right side of the cell. The actions, the viral actions, project to the left side of the cell, where host defenses are organized. And one of the key set of defenses is an antioxidant system based on the synthesis and processing of glutathione, a tripeptide molecule. Its abbreviation, once again, is GSH. Glutathione is utilized by glutathione peroxidase, and glutathione peroxidase 4 serves as a major antioxidant protection against viral ferroptosis. Once again, GPX4 chemically reduces reactive oxygen species as well as peroxidated lipids, upstream drivers of ferroptosis. What are glutathione peroxidases? Glutathione peroxidases, abbreviated GPX, are extensively studied for their indispensable roles in eliminating reactive oxygen species by catalyzing the chemical reduction of hydrogen peroxide or lipid peroxides to prevent cell damage. In looking at this diagram, we are visualizing a metabolic map of viral ferroptosis. It is a partial map, but comprehensive in that it connects many viruses to the common processes of the NRF2 GPX4 anti-ferroptosis signaling axis. It is now important to connect this comprehensive map of viral ferroptosis to a broader set of information that pertains to the pathogenesis of COVID and long COVID. To do that, it's useful to start with an analogy, taking this metabolic map of viral ferroptosis and comparing it to a map of the Earth, that of a globe. We are in the early stages of linking ferroptosis pathways and viral pathogenesis to a wealth of information that has been obtained from the study of COVID and long COVID. So I would like to bring in this paper, published also in 2025, Integrative Multi-Omics Framework for Causal Gene Discovery in Long COVID. And there is a very good graphical abstract that I encourage you to consult. So, reading from the paper, our study has identified 32 potential causal genes for long COVID, of which 19 have been confirmed by the existing literature, providing support for the effectiveness of our findings. The remaining genes represent promising candidates for follow-up experiments. Among these candidates, we identified genes that act as risk or protective factors, as well as network driver genes that regulate and stabilize the disease network's structure. Enrichment analyses revealed important biological pathways in long COVID, including SARS-CoV-2 infection, viral carcinogenesis, cell cycle regulation, and immune response mechanisms. Using the identified potential causal genes, we clustered long COVID patients into 3 distinct subtypes with different symptom profiles, establishing a foundation for personalized diagnostic and therapeutic approaches. This work represents a significant step towards customized management and treatment strategies for long COVID, ultimately improving patient outcomes. To facilitate the application of our framework, we developed a web application, a Shiny app, that allows users to generate gene lists by adjusting parameters for direct and network-based causal approaches. This tool provides researchers and clinicians with an accessible platform to explore parameter variations and analyze their data, enhancing the reproducibility of our findings. In this regard, it is very important to insert the NRF2 GPX4 anti-ferroptosis axis into this analysis. And upstream of NRF2 and GPX4 is melatonin, and that connects to the major theme of this YouTube series, the antiviral properties of sunlight, the production of melatonin, as well as vitamin D and its hydroxylated derivatives, calcifediol 25 vitamin D and calcitriol 125 vitamin D. Among the 32 causal genes that were identified for long COVID, one of the top high-risk genes for long COVID was identified to be the CREBBP gene, which is located on chromosome 16. This gene encodes the CREB binding protein, CBP, a crucial co-activator in histone acetyltransferase that regulates gene expression, cell growth, and differentiation. It acts as a scaffold to control chromatin structure and is essential for normal development and memory. Mutations in CREBBP cause Rubinstein-Taybi syndrome, while fusions are linked to leukemia and various cancers. A key question now is whether the key players that are upstream of viral ferroptosis, agents that are being attacked by viruses, such as the cysteine glutamate antiporter, also known as XCT, or the subunits to this transporter complex affected in long COVID. Similarly, are key players involved in the anti-oxidation, anti-ferroptosis axis, NRF2, GPX4, and an inhibitor of NRF2, KEAP1, KEAP1. Do any of these genes show up in the integrative multi-omics analysis framework for causal gene discovery in long COVID? So as I mentioned, this map of viral ferroptosis is comprehensive, but it's an early map. So another analogy, this is like the early globes of the world, and this shows the earliest extant globe of the world, which was constructed in 1492. So we can anticipate that these types of diagrams, as they become linked to COVID and long COVID databases, will become much richer in detail in a manner similar to the refinement of early globes of the Earth. The use of globes helps remind us of the geographic aspects of viral infections. For certain respiratory viruses such as influenza and COVID, there is a clear seasonality, and the susceptibility of cells and tissues to respiratory viral infections is tied to these anti-ferroptosis pathways. Now, there's a takeaway concept from this paper, critical role of ferroptosis in viral infection and host responses. And that is that there is a high consistency of viral attacks that are focused on antioxidant pathways. And some of the key players which are attacked by different viruses are the cysteine glutamate antiporter, also known as XCT complex. It produces glutathione, abbreviated GSH. Glutathione is the substrate for glutathione peroxidase 4, an essential enzyme needed to detoxify the cell from reactive oxygen species as well as peroxidated lipids, the upstream drivers of ferroptosis. In addition, certain viruses attack a transcription factor, its expression, NRF2, NRF2, which is upstream of the expression of GPX4. So melatonin is a probable primary defense agent against viral ferroptosis. And as we have discussed, melatonin is produced inside cells, particularly mitochondria, in response to near-infrared light. There is, of course, a strong diurnal rhythm of sunlight exposure, and thus the near-infrared light needed to produce melatonin within mitochondria. In addition, there is a circannual rhythm, a yearly rhythm tied to the seasons, with reduced sunlight during winter months, resulting in a deficit in the production of melatonin. And these issues have been analyzed in this paper, A Geographical Approach to the Development of Hypotheses Relating to COVID-19 Death Rates. And what the authors report is a latitudinal dependence in seasonal COVID mortality. In other words, the seasonal mortality of COVID, if you analyze the rates of COVID deaths, is a function of latitude within countries like the US or the UK, you find a latitudinal dependence with greater numbers of deaths at higher latitudes during winter months. This is, of course, linked, it's a correlation with reduced sunlight and by inference, reduced near-infrared light. And these issues are discussed by Professor Roger Schwelt at UC Riverside on the MedCram channel. And this YouTube, Sunlight for Hospitalized COVID Patients, discusses the treatment of a COVID patient, a patient of Dr. Schwelt, and this patient was on oxygen support. He was taken outside to receive sunlight, and then within 24 hours, his respiratory status improved, and he was able to be discharged several days later. So I refer you to that YouTube for the details of this case study. Sunlight treatment for hospitalized COVID patient, outcome and implications. And Professor Schwelt does go into latitudinal dependence of COVID mortality in this set of YouTubes. If you go to PubMed and search for sunlight exposure in COVID, there's 250 papers on this topic. And one of these papers, Sunlight Exposure Increased COVID-19 Recovery Rates, a study in the pandemic area of Indonesia. And this directly connects to the case study of this COVID patient who was wheeled out of the pulmonary ward in this hospital and treated with California sunlight. I would like to add an additional analogy, that of an Achilles heel in these processes, and that is GPX4, glutathione peroxidase 4. Its expression can be downregulated by a variety of processes. And in the case of COVID, because of ischemia, there is a good chance that the promoter region for this gene becomes methylated, leading to gene silencing. And gene silencing is when the transcription of the gene is silenced, meaning no proteins can be generated from the silenced gene. And this would occur sporadically within a COVID patient's body in response to certain stimuli. And one of those is likely to be ischemic reperfusion injuries. I would now like to do a quick deep dive from the epidemiological data which was mentioned in the YouTube Sunlight for Hospitalized COVID Patients, namely the latitudinal dependence in COVID mortality, and bring that down to the subcellular level, particularly mitochondria and peroxisomes. So in this YouTube, Professor Schwelt was discussing this key paper, Ultraviolet A Radiation and COVID-19 Deaths in the USA with replication studies in England and Italy. And this paper was published in 2021. This epidemiological data needs to be connected to ferroptosis biomarkers within cells, particularly cells that have been treated with sunlight or near-infrared light. And that brings us to this paper on the right. Near-infrared light exposure triggers reactive oxygen species to downregulate inflammatory cytokines induced by SARS-CoV-2 spike protein in human cell culture. Now, this paper is complex, but it comes to some very precise conclusions. So let me first read the abstract. The leading cause of mortality from SARS-CoV-2 is an exaggerated host immune response, triggering cytokine storms, multiple organ failure, and death. Current drug and vaccine-based therapies are of limited efficacy against novel viral variants. Infrared therapy is a non-invasive and safe method that has proven effective against inflammatory conditions for over 100 years. However, its mechanism of action is poorly understood and has not received widespread acceptance. We herein investigate whether near-infrared light, NIR light, exposure in human primary alveolar and macrophage cells could downregulate inflammatory cytokines triggered by the SARS-CoV-2 spike protein, or lipopolysaccharide, LPS, and via what underlying mechanism. Our results showed a dramatic reduction in pro-inflammatory cytokines within days of near-infrared light treatment, while anti-inflammatory cytokines were upregulated. Mechanistically, near-infrared light stimulated mitochondrial metabolism, induced transient bursts in reactive oxygen species, and activated antioxidant gene transcription. These, in turn, downregulated reactive oxygen species and inflammatory cytokines. That's the therapeutic pathway. A causal relationship was shown between the induction of cellular reactive oxygen species by near-infrared light exposure and the downregulation of inflammatory cytokines triggered by SARS-CoV-2 spike protein. If confirmed by clinical studies, this method would provide an immediate defense against novel SARS-CoV-2 variants and other inflammatory infectious diseases. Now, there's a graphical abstract in the upper right. Let me walk through that slowly. So panel A, there's a near-infrared light source radiating near-infrared photons. They strike mitochondria. That's K1. In response to the near-infrared photons, the mitochondria release reactive oxygen species, ROS. One of those is hydrogen peroxide, H2O2. Let us pause here for a sidebar comment. At first sight, this is surprising. Near-infrared light is supposed to be therapeutic, yet what the authors have demonstrated is that near-infrared light, by striking mitochondria, produces a very rapid release of reactive oxygen species from the mitochondria. That's a key new piece of information. What happens is the cell reacts to this reactive oxygen species burst, which occurred in response to near-infrared light, and it adapted. It turned on anti-inflammatory pathways to deal with the reactive oxygen species. And by doing so, prepared the cell to undergo a shift in its cellular trajectory. Instead of being in a pro-inflammatory state, with repeated near-infrared exposures, the cell, by adapting to the reactive oxygen species, became resistant to the reactive oxygen species. And we will see one of the key enzymes which was upregulated in response to the near-infrared light. The net result of this is that pro-inflammatory cytokines are suppressed, and that's shown on the right, cytokine secretion being suppressed with the X. Now moving to panel B, and that's the lower part of the inset of the graphical abstract. This lower sequence shows near-infrared photons being delivered to cultured cells for various amounts of time. 5 minutes, 10 minutes, 15 minutes, and 20 minutes. There's a typo in that legend. 1 should actually be 15. And what they're showing is the amount of ROS, reactive oxygen species, is going up in rough proportion to the duration of the near-infrared photons being delivered to the cells. So K2. So once the cells have been dosed with near-infrared radiation, have produced reactive oxygen species in response to that, then they analyze the amount of cytokines in the cells. And what they found was an optimum duration of time to reduce the pro-inflammatory cytokines. And that optimum time was 10 minutes. So once they had decided on using 10 minutes of exposure time for near-infrared irradiation of their cell cultures, they then proceeded to utilize both spike protein as well as another agonist of the toll-like receptor 4, lipopolysaccharide, abbreviated LPS. LPS is commonly used in cell cultures and rodent studies to provoke rapid inflammation. So using the 10 minutes of near-infrared illumination as well as the LPS trigger for cell inflammation, they conducted an experiment which is shown, the results of it, just to the left. And they're looking at CAT, which stands for catalase. Catalase is a key enzyme which degrades hydrogen peroxide. And catalase is primarily located inside peroxisomes, which are often adjacent to mitochondria, which produce hydrogen peroxide. Now that's a lot of verbiage. Let me go to the experiment. They're looking at the amount of gene expression for catalase, not the protein, not its biological activity. They're looking at the amount of gene expression, amount of RNA. So in control cultures, there's a certain amount of catalase production. And this is the RNA which will make protein, catalase protein, which will be stored inside peroxisomes, ready to deal with hydrogen peroxide. So in response to lipopolysaccharide, that's the blue bar in this histogram, note that this toll-like receptor agonist, which provokes cells into an inflammatory condition, depressed catalase levels. And this is potentially bad because with reduced catalase, the ability of the cell to degrade hydrogen peroxide is reduced, leading to conditions that could promote ferroptosis. Now the red bar illustrates the therapeutic benefits of near-infrared light. These cells were treated with a combination of lipopolysaccharide and 10 minutes of near-infrared light. Notice that the catalase levels were boosted in response to this. So the result of lipopolysaccharide is to stimulate toll-like receptor 4, which sets off a pro-inflammatory cytokine cascade. There's also a production of reactive oxygen species in cells in response to LPS. Normally, in the blue bar, those cells, catalase messenger RNA expression was depressed, but in the cell culture which was stimulated with near-infrared light, the opposite happened. Catalase levels went up, and this again is the messenger RNA, the measure of gene expression. So this deep dive into the levels of catalase messenger RNA expression in response to a pro-inflammatory agonist, which is found on bacterial surfaces, bacterial cell wall surfaces. Utilizing near-infrared light, one produces a state which is more advantageous for the cells to degrade hydrogen peroxide, reduce this type of reactive oxygen species, and thus reduce the susceptibility of the cells to ferroptosis. One may ask, how significant is LPS in the context of cellular pathophysiology? LPS is a common component of the cell wall that is beyond the cell membrane of the bacterium of gram-negative bacteria, which include E. coli. In E. coli, LPS constitutes 80% of the molecules on that outer surface, the cell wall of the bacterium. So let me put the context of ferroptosis in a tissue and organismic context. If you have bacteria on the surface of an epithelium, such as the gut or the lungs, and LPS is being liberated, setting off toll-like receptor 4, pro-inflammatory cytokines, reactive oxygen species, if the cells are not able to process the reactive oxygen species, this will lead to peroxidation of lipids, setting the stages for ferroptosis, or programmed cell death. These will lead gaps in the epithelium, allowing the bacteria to penetrate into the deeper tissues of the organ. And this is what's called sepsis. And sepsis is a common pathophysiological state found in advancing progressive COVID patients. Now, shifting the discussion, the narrative back to the COVID patient who was being treated with sunlight. They wheeled this patient who had a pulmonary infection of COVID-2. His condition was worsening. They moved him to sunlight, and within a day, his pulmonary functions improved. And within several days after that, he was discharged. How could sunlight produce such a rapid change? Well, if we look at this paper, which was published in 2023, we go all the way down to the level of catalase messenger RNA being upregulated in response to near-infrared photons. As proteins, catalase proteins, are upregulated, these cells will be able to resist reactive oxygen species more effectively. And that would be of major benefit for somebody who is suffering from a COVID-2 infection of the lungs or the gut, because if catalase is deficient, reactive oxygen species will set off a cascade of results leading to ferroptosis. And that's shown at the lower left of this slide. That needs to be prevented. And this is where sunlight and near-infrared lights now appear to be a promising way of arresting the pathophysiology that's taking place, which could lead to sepsis. And so, again, reading from the abstract of this paper, if confirmed by clinical trials, this method, using near-infrared light, would provide an immediate defense against novel SARS-CoV-2 variants and other inflammatory infectious diseases. So, takeaway concept. We have seen that there is a high consistency of viral attacks that are focused on antioxidation pathways. And some of the key players inside cells that provide this antioxidant capacity are the cysteine glutamate antiporter, also known as XCT, glutathione, abbreviated GSH, NRF2, the key master anti-inflammatory antioxidative transcription factor, which upregulates genes such as GPX4, glutathione peroxidase 4. And we've seen how researchers emphasize this is an essential enzyme to resist and degrade reactive oxygen species and to chemically reduce peroxidated lipids, which are the upstream drivers of ferroptosis. Now, adding to this list is catalase, the enzyme which is placed in peroxisomes, which are located next to mitochondria. And catalase, its role is to convert hydrogen peroxide into water and neutralize it, neutralize the hydrogen peroxide. Now, the importance of peroxisomes in COVID and long COVID has only become apparent in the last couple of years. I would like to draw your attention to this blog from the Brain Inflammation Collaborative, Oxidative Stress in Long COVID and ME/CFS, Myalgic Encephalitis, Chronic Fatigue Syndrome, the Mitochondrial Peroxisomal Vicious Cycle, Oxidative Stress and Mitochondrial Dysfunction, Dry Fatigue and Brain Fog in Long COVID and ME/CFS. Discover the peroxisomal vicious cycle, proven ways to boost glutathione, NAC, N-acetylcysteine, coenzyme Q, and NRF2, boosting NRF2. And as we've been discussing, one way to boost NRF2 is with melatonin, which can be produced by near-infrared light. Now, before we close this YouTube, I'd like to bring up some of the supporting new information behind these conclusions. We have just discussed how catalase depletion can set up cells for ferroptosis. That was an enzyme-centric analysis. Now we're going to talk about a paper with an organelle-centric analysis focusing on peroxisomes in macrophages. This paper was published in 2025 in Science. Macrophage peroxisomes guide alveolar regeneration and limit SARS-CoV-2 tissue sequelae. Conclusion. This study suggests that severe respiratory viral infections can reduce peroxisome biogenesis and promote peroxisome degeneration in response to raised interferon levels. Our findings reveal that peroxisomes act as essential regulators of macrophage-mediated lung inflammation resolution and tissue regeneration following viral injury. This is the physiology of remission. Thus, peroxisomal dysfunction in macrophages contributes to the development of severe acute morbidity and chronic tissue sequelae of post-COVID-19, i.e. long COVID. Targeting peroxisome biogenesis or enhancing peroxisomal metabolic function represents a promising therapeutic approach for mitigating the long-term consequences of respiratory viral infections, with potential for improving health outcomes for patients with PASC, post-acute sequelae of COVID. I will first read the legend to this graphical summary of this paper and then tie it back to concepts that we have been discussing earlier in this YouTube. The takeaway concept of this paper is that it is possible to protect peroxisomes in both pulmonary macrophages as well as the epithelial cells of alveoli using a drug known as 4-PBA. So I will now read the legend to this graphical abstract and then go in and define key acronyms for better definition of these concepts. Model of peroxisome function in macrophage-mediated alveolar regeneration after viral injury. Peroxisome activity in alveolar macrophages is essential for AT2 self-renewal, These are a type of alveolar epithelial cells, as well as alveolar repair following viral infection. Dysfunction of macrophage peroxisomes causes excessive inflammasome activation and promotes dysplastic, means abnormal cells or abnormal development. KRT8, we'll define that shortly, transitional cells accumulation, resulting in chronic tissue sequelae post-acute infection. We're talking about PASC. 4-PBA treatment facilitates peroxisome biogenesis, the growth of peroxisomes, in macrophages to enhance alveolar regeneration after respiratory viral infection. So what is 4-PBA? It stands for 4-phenylbutyrate. And butyrate is a major stimulus for the gut lining of vertebrates. And people take psyllium fiber as a natural source of butyrate. It is fermented by bacteria in the gut, releasing butyrate, which stimulates epithelial cells, enterocytes of the gut, to differentiate and form a protective coat of mucin on the surface of the epithelium. So 4-phenylbutyrate is a derivative of butyrate, butyric acid. This chemical acts as a chemical chaperone that protects against peroxisomal injury, specifically preventing the loss of peroxisomal proteins like PM, P40, and catalase, which we have been discussing, the enzyme that degrades peroxide. So this chemical prevents the loss of peroxisomal proteins caused by inflammation. It helps maintain peroxisome function in central nervous system cells and is linked to the stimulation of peroxisome proliferation via PEX11A, 11 alpha. What are AT2 cells? This stands for alveolar type 2 cells. And then this paper is a useful reference. Alveolar epithelial type 2 cells in acute lung injury and acute respiratory distress syndrome, ARDS, spatial temporal fate and regulation. They are talking about the regeneration of the alveolar epithelium after acute lung injury and acute respiratory distress syndrome, which is inflammatory. Alveolar type 2, AT2 cells and macrophages form a critical interactive niche in the lungs, where AT2-derived GM-CSF, which stands for granulocyte macrophage colony stimulating factor, this growth factor is crucial for the development and maintenance of alveolar macrophages, the innate immune cells which patrol the surface of a lung, and phagocytose and digest pathogens like viruses and bacteria. Macrophages in turn regulate these alveolar cells, AT2 cells, and their cell proliferation, their repair, and their fate after injury, with alveolar macrophages, these are the macrophages in the alveoli, sacs, often acting in a M2 polarized, that's a repair phenotype, to support remodeling, i.e. regeneration. Now, what are KRT8 positive cells? That's keratin 8 positive. Sidebar comment. Clearly, these acronyms are complex and difficult to articulate. However, it is a way that biologists, cell biologists, immunologists, developmental biologists, neurobiologists have been forced to adopt to compress the amount of syllables and space in publications. So it's difficult to remember all of these acronyms, so I find it useful to go to AI and ask what is the definition of this acronym. That's how I am generating these insets. So keratin 8 positive cells are a transient transitional cell type derived from activated alveolar type 2 progenitors. So the AT2 cells are part of the alveolar epithelium. When the alveolus is injured, they are activated and they divide and form this transient transitional cell type, which is called keratin 8 positive cell. So these cells play a key role in lung injury repair by differentiating into alveolar type 1 cells, AT1 cells. These cells often show signs of senescence and can aberrantly persist in pulmonary fibrosis. They're talking about the keratin 8 positive cells acting as a source of disease progression. So I have added a red arrow between KRT8 positive cells, keratin 8 positive cells, to the site where they're illustrated in the summary diagram. And these cells persist in PASC, leading to pulmonary fibrosis and loss of vital capacity. That's the term pulmonologists use for how much respiratory surface, functional respiratory surface, the lungs have. Now I'm going to tie all of these pieces together, so it's going to make a complicated slide. So bear with me as I move through it. In this YouTube, we have talked about latitudinal dependence in COVID mortality, and we have discussed reasons why near-infrared light may be the source of this. And near-infrared light, as we've seen, is able to boost a key enzyme, catalase, which degrades peroxide. Peroxide is generated by active mitochondria. And if there's inadequate degradation, inactivation of this peroxide, it will peroxidate lipids, and peroxidated lipids will trigger a programmed cell death called ferroptosis, which also we've discussed in this paper. So this is part 2 of this YouTube. Part 3 of this series of concepts, we've talked now about macrophage peroxisomes, how protecting those peroxisomes during COVID and long COVID, you need those macrophages in order to guide alveolar regeneration and limit SARS-CoV-2 tissue sequelae, namely pulmonary fibrosis and loss of lung function. So takeaway concept, this is 4. Peroxisome deficits in macrophages, pulmonary macrophages, are linked to pulmonary dysfunctions in long COVID. And so the authors have illustrated that this 4-phenylbutyrate compound is a potential therapeutic agent to protect peroxisomes during COVID and to help regenerate peroxisomes in PASC. So now how does this tie to the patient that was described in the Medcram YouTube, sunlight as a treatment for hospitalized COVID patients? So this patient, who had pulmonary dysfunction from a COVID-2 lung infection, was wheeled out into an atrium and allowed to be treated with sunlight for about an hour. And within a day, his pulmonary function improved, and several days after that, he was discharged from the hospital. So Professor Roger Swelt describes this as a surprising and unexpectedly dramatic turnaround of his progression. So how does sunlight connect into this patient's recovery? Well, we've been talking about how near-infrared light can boost catalase expression even in the presence of inflammatory agents such as lipopolysaccharide, which is being produced in the lungs in response to bacterial infections. And it should be pointed out that respiratory viral infections often create immune dysfunction, allowing bacteria to proliferate. You end up with what's called a polymicrobial infection, which includes both viruses and oftentimes bacteria. These bacteria could produce this lipopolysaccharide. So near-infrared light, either from the sun or from therapeutic red light, near-infrared light boxes, one way that they could potentially benefit pulmonary patients in these acute conditions is to boost catalase levels. And catalase helps protect the cells from peroxidation of lipids, which will trigger cell death, ferroptosis. So in closing, I would like to draw your attention to several YouTubes posted on this channel which deal with ferroptosis being driven by GPX4 silencing. And this occurs when the promoter region for the gene that encodes GPX4 becomes hypermethylated. So gene silencing is a major histopathological process as cells lose the function of certain genes like GPX4 or vitamin D receptors. These receptors and enzymes that carry out key anti-inflammatory antioxidant processes are no longer able to be synthesized. This is because the RNA from these silenced genes is not being produced. So in this YouTube series, I review a key paper from 2024, GPX4 promoter hypermethylation induced by ischemia reperfusion injury regulates hepatocytic ferroptosis. Ischemia reperfusion refers to the lack of blood flow, often resulting from a blood clot or surgery or ligation, and then opening the blood vasculature and allowing blood to return. This reperfusion brings oxygen into the tissue and the cells are not prepared for it. There's a lot of reactive oxygen species being produced. This triggers ferroptosis. And so there is a key weak spot, and that's the promoter region for the GPX4 gene, which can become hypermethylated. And GPX4 plays key roles in suppressing bacterial infections and polymicrobial sepsis. So GPX4 in bacterial infection and polymicrobial sepsis, involvement of ferroptosis and pyroptosis. These are two different forms of programmed cell death. So if you look on PubMed for the intersection of the concepts of ferroptosis and sepsis, there's 548 results. Note that in the histogram, these are very recent papers. As I said earlier in this YouTube, the concept of ferroptosis as a histopathogenesis mechanism is relatively new, and it has been driven by the COVID pandemic era. Fortunately, there are newly recognized ways to suppress ferroptosis. One, by stimulating vitamin D receptors with 1,25-dihydroxy vitamin D metabolites such as calcitriol, 1,25 vitamin D3, or paracalcitol, 1,25 vitamin D2. In addition, one can boost 25-hydroxy vitamin D levels. These are calcifediol levels. And calcifediol is what doctors measure when they're making a, quote, vitamin D test on someone's blood. Again, this is always ambiguous terminology that needs to be clarified. Vitamin D is a nutrient. It's inactive. That's not the therapeutic that will suppress ferroptosis. You need to go to the active vitamin D receptor agonist, 25D or 125D, in order to boost NRF2, which in turn boosts GPX4, which can then suppress the reactive oxygen species and peroxidated lipids that are driving ferroptosis. The other way to suppress ferroptosis is through near-infrared light, either from sunlight or a therapeutic light box that produces near-infrared light. And the hormone that will be produced from the near-infrared light is melatonin, being generated within mitochondria. The advantage of near-infrared light, it can penetrate many centimeters into the body. Melatonin upregulates NRF2, the master anti-inflammatory antioxidant transcription factor, which in turn upregulates the expression of the antioxidant enzyme, or critical enzyme, GPX4, which is needed to suppress reactive oxygen species and peroxidated lipids, and thus serve to suppress ferroptosis. Finally, I would like to emphasize that cells that have had their GPX4 promoter hypermethylated are going to be in a very difficult situation, because no matter how much melatonin or vitamin D receptor agonists are given to the cell, even if you upregulate NRF2, the GPX4 gene will have been silenced, and thus, without this critical enzyme, the cell is set up for ferroptosis. In previous YouTubes on this channel, we have discussed research into DNA methyltransferase inhibitors such as 5-AZA2, which in animal models show promise in suppressing the methylation of genes. And with time, demethylation can be expected. That is a topic for future YouTubes. So with that, if you would like a PDF slide summary of this particular YouTube, please navigate to this Google Docs folder highlighted in green at the bottom of the slide. Inside this Google Docs folder, there are subfolders. If you'll navigate to download first, you'll be able to find this PDF slide summary of this YouTube most quickly. The other folders contain references to ferroptosis and COVID, ferroptosis and ferritinophagy, the dysfunction or destruction of ferritin complexes releasing iron. This is a primary driver of ferroptosis. And finally, a set of references on GPX4 gene silencing. You are welcome to download all of the materials in these folders.